Hello everyone, myself Sneha and I will be discussing about factors affecting human disease frequency. Before coming to the main topic, a simple question comes that what is disease frequency? Measures of disease frequency are used to describe how common an illness or other health event is with reference to the size of population at measure of a time. Next question is why measuring of disease frequency is important. By measuring the frequency of a disease in a population and identifying how the disease frequency may differ over time or among subgroups are important steps in discovering potential causes of a disease and determining effective methods for prevention and care. We can see in the slide is that there are various factors which can affect disease frequency. We know that disease can be spread from individual to individual by carriers. So carrier is one such factors that harbors the pathogen and spreads it to other individual. Some pathogen, for example cold and flu viruses, are transmitted through air. Next is population pattern, like exactly where and how people live and interact with others. And densities, like how many people are present within a defined area, are also important in the spread of disease. People living in close proximity, such as in city, are more likely to spread infection. Many times, environment plays a vital role in the spread of disease that can be caused due to pollution and hygiene. And next is genetic inheritance of disease, like cystic fibrosis, sickle cell anemia, alpha and beta thalassemias, etc. It also depends on gender. Many times female only get affected by the disease and sometimes only male. Other factors we can see here in the factors affecting Hardy-Weinberg equilibrium. First, we need to know that what is Hardy-Weinberg principle. This principle states that allele and genotype frequencies in a population will remain constant from generation to generation in the absence of other evolutionary influences. But due to many factors, Hardy-Weinberg principle gets deviated and leads to change in frequencies. First factor is non-random mating, in which individuals with similar phenotypes mate with one another, and any departure from random mating upsets the equilibrium distribution of genotypes in a population and also leads to change in disease frequency. Next is mutation. It is the alteration in nucleotide sequences of genome. Simply, it will change allele frequency and hence genotype frequency. Next is selection. So natural selection, when individuals with certain genotypes are more likely to survive and reproduce and then pass their allele to the next generation. The allele will spread in the population and may become fixed. Smaller population is more susceptible to mechanisms such as genetic drift. Random events that alter allele frequencies will have a much larger effect when the gene pool is small. Also, disease can spread easily. Next is gene flow. It will change gene frequencies by bringing in more copies of an allele already in population or by bringing in a new allele that has arisen by mutation. Migration will spread the allele or disease and leads to change in frequencies. There are two basic equations in Hardy-Weinberg principle. The first equation is p square plus q square plus 2pq equals to 1 and p plus q equals to 1. P is the frequency of dominant allele in population. Q is the frequency of recessive allele in population. Square is the percentage of homozygous dominant individual. Q square is the percentage of homozygous recessive individual, and 2pq is the percentage of heterozygous individual. Now I would like to discuss a question in which Hardy-Weinberg equation can be used. So the question is, in United States approximately one child in 10,000 is born with phenylketonuria. So phenylketonuria is a syndrome that affects the individual's homozygous for recessive allele. Recessive allele can be denoted by small a. Now the first part of the question is, calculate the frequency of this allele in the homozygous. So small a can be denoted as recessive allele, and in Hardy-Weinberg equation frequency of recessive genotype can be denoted as q square. Q square can be calculated as 1 by 10,000 and the value of Q is 0.01. Next part of the question is, calculate frequency of normal allele. Capital A represents normal allele and is also represented by P in Hardy-Weinberg equation. We can put the equation P plus Q equals to 1 here. So P can be calculated as 0.99. The third part of the question is, calculate the percentage of carriers of the trait within the population. Heterozygotes are the carriers of the trait and are represented by 2pq. So from previous parts we got the values of p and q, and now we can put the values here. So 2pq can be calculated as 0.0198. So here I would like to conclude that Hardy-Weinberg equation can be used to evaluate real genetic structure of a population over time. Thank you.